Okay. So before we get started, just a couple of housekeeping items. The first of which is this week we'll have another quiz. So that's going to be on Thursday. That should be over Elephants on Acid chapter 4, right? And then whatever we cover this week. And my goal is to get through all of chapter 4 in the text this week. So finish up the Torah of the Sal, and then move on to some better things. This week in laboratory, we'll be doing laboratory number 6, which is microscopy in the Sal. So please read that before you come. It'll certainly help. We also need to talk a little bit about the data that we collected last week for the owl pellets. So we'll start the lab by doing that, and then move on to the actual lab content. Okay, so we left off, excuse me, we left off talking about the endoplasmic reticulum. And you'll recall that there were two types. There was a rough endoplasmic reticulum, and there is a smooth endoplasmic reticulum. What's the primary difference between the two? The structural difference between the two? Yeah, Mac.
One has more ribosomes than the other, which is the rough.
Right.
More ribosomes than the smooth.
Exactly right. So it's a difference in the amount of ribosomes. And really, smooth ER has a total of 0 ribosomes. So no ribosomes in the smooth ER. The rough ER has all of the ribosomes, right, if it's embedded in membrane. And because the rough ER has ribosomes, that means its function is what? What does it do?
Synthesizes protein.
Proteins, exactly right. So it's going to make proteins. Now remember, please, that if the rough ER is making proteins, those proteins are primarily for secretion. They're not used within the cell. Generally, they're exported out of the cell. The smooth ER is going to make fats and sugars, and it's going to detoxify stuff, right? Now the fats are important for making more membrane, and there's lots and lots of membrane inside the cell and outside the cell. The question is, how does something get from the endoplasmic reticulum to elsewhere in the cell? And the answer to that question is something called a vesicle. And the vesicle is just a little bubble of membrane that has some endoplasmic reticulum product. So if a protein is synthesized, a portion of the endoplasmic reticulum buds off and becomes like a little bubble around that protein, and it carries it elsewhere in the cell. Now generally, when a protein is made by a ribosome, it's not the completed product. It needs to go somewhere else to be finished. So this is a scanning electron micrograph, and this is also a scanning electron micrograph, and it shows you the difference between the rough ER and the smooth ER. So the rough ER, you'll notice, is a series of lines that has little dots on either side of the lines. That line is membrane, and the dots on either side of the line, excuse me, are the ribosomes. So you can see that it's super wavy, lots and lots of lines in the rough ER, with lots and lots of ribosomes embedded in it, which means that the potential for protein production just within the rough endoplasmic reticulum is very high. This is a scanning electron micrograph. I'm sorry, it's a transmission electron micrograph of the smooth ER. And you'll notice that it's lots more irregular. If that even makes sense. It's irregular, right? This has a very organized linear shape to it. This is really, really random looking. But more importantly, it doesn't have any little bumps associated with it or dots associated. There are no ribosomes in the smooth ER. And then this shows you little vesicles budding off of the rough ER that are carrying some product. Here it's going to be a protein product to the next organelle we're going to talk about, which is the Golgi apparatus. So the Golgi apparatus has a similar construction to our endoplasmic reticulum. And it's a series of flattened stacks of membrane. So it's a highly convoluted membrane sac. I'm just going to say highly wavy, because I don't know that convoluted makes much sense to you guys. But it's a highly wavy membrane sac. And those waves of the membrane organize themselves into things called cisterns.
How do you spell that?
Cistern, C-I-S-T-E-R-N-S. I think that's how I spell it. I don't know if that's how it's correctly spelled. So it's a highly convoluted sac of membrane. That's what we have pictured right here. So you can think about it just like a bunch of stacks of membranes squished together. And those extensions right here, the ends of them are called cisterns. Do you guys know, like, outside of biology, what a cistern is? If you guys have a cistern on your house, what does it do? Collects. Oh, is that not what you said? Did you not say collect? I'm putting words in your mouth, Adam. Adam says it collects water, and Adam's exactly right. That's what a cistern does. If you have a large cistern by your place, it's a water collection bin, basically. Cisterns in the Golgi apparatus do the same thing. They collect things from elsewhere in the cell. And the job of the Golgi apparatus is to take all of those things that are made in the cell and to finish them up, and then to distribute them to where they need to go. So, if we were to look at the cell as like a big, I don't know, manufacturing sort of junta kind of thing, what happens, right, is the nucleus is in control of all of the stuff that's being made. So it's determining, if it's IKEA, right, it's corporate headquarters, and it's saying we need to make more couches. So it sends that message to a factory, and the factory starts making the parts for these couches. But that's not the end of it, right? It's not just a bunch of parts. Those parts need to be taken somewhere, put in the right boxes, and then shipped out to the stores. And that's what the Golgi does, right? The Golgi takes all those disparate parts, puts them together, and then ships them out to wherever they need to go. It's like a distribution center, really. That's what the Golgi apparatus does. The endoplasmic reticulum is that factory that's actually making the parts, and the Golgi apparatus is taking all those parts, kind of putting them together, and then shipping them out, okay? The question is, how does it do that? And there are two models for how it is that the Golgi works that I'm going to want you to be familiar with. So, let me write function really quickly. The function of the Golgi apparatus is to receive, complete, and distribute cellular products. So there are these cisterns in the Golgi apparatus, and we know that cellular products are going to kind of move through those cisterns, and each cistern has some specific job as if it's kind of like an assembly line. But there are two different models that describe how it is that can function. And we're going to call the first model the static cistern model. And what the static cistern model says is a product is going to be received by the Golgi apparatus. It's going to move into a cistern, and that cistern is going to do some specific set of tasks with it. Once that cistern is done with that product, the product is going to move to the next cistern, and then that cistern is going to do some stuff with it. And then it'll move to the next cistern. Let me model that really quickly, or diagram that on the board. So if this is our Golgi apparatus, I'm just going to draw one half of it. Let's say that it consists of 5 cisterns, right? What happens is that this is called the cis base right here. And the cis base is always directed towards the endoplasmic reticulum. And this is the receiving area. for the Golgi apparatus. What's going to happen is the vesicle is going to come along, and it's going to bind to the membrane of the Golgi apparatus, and it's going to release its product inside the Golgi. And then that product, I'll draw that. I'll say that this is a little vesicle that's arrived from the endoplasmic reticulum. It's going to release a little protein into this first cistern. So that protein ends up right here. Now, the static cistern model says that that protein's going to stay there until this cistern has had its way with it and it's finished up all the things that it needs to do. Now, that could be taking parts of the protein out and resplicing it, or it could be inserting new parts into the protein. Whatever needs to happen, this first cistern is going to do some of that stuff, right? And it's able to do that because there's a specific protein composition embedded in the membrane of this cistern that catalyzes specific reactions. Once all those reactions have been completed, then that protein is going to move from this cistern to the next. And that protein is going to live in here until this cistern is done with it. And this cistern has proteins embedded in its membrane as well, but they're different than this membrane. So there is a unique identity to each sequential cistern. And then this protein just bounces between cisterns until eventually it's finished, and it's going to bud off the transphase, which is up here at the top. And off the transphase, this completed protein is ready to go wherever it's needed. If it's a digestive enzyme, it's going to be taken to the plasma membrane and secreted out into the cell, perhaps. Okay, so that's the static cistern model. Does that make sense? The salient point here is it's the protein that's moving between cisterns, and the cisterns are remaining static. They're not moving at all. Alternatively, there is the cisternal maturation model.
That's the second one?
It is. Number 2 is the cisternal maturation model. So if I was to just say the word mature, what does that mean to you guys?
Fully developed.
Okay, to develop. So if I said my daughters are maturing, what would that mean? They're changing a little bit, right? Maybe they're becoming more responsible. Evidence suggests contrary, but like in theory, that's what they're supposed to do, right? They're supposed to get older, they're supposed to mature, and they're supposed to become a little bit more responsible. So that's what's happening with the cisternal maturation model as well. It's not necessarily the product that's moving, it's actually the cisterns that are moving along. And as the cisterns move, their protein composition changes, and they're able to accomplish different tasks as they move along the surface of the Golgi apparatus. And so a diagram of that would look something like this. Okay, so again we have our cis phase, which is directed towards the endoplasmic reticulum, right? And we have a trans phase up here, and the goal is to get the product from the cis to the trans phase and then ship it out. So we receive a vesicle from the endoplasmic reticulum. That's carrying a protein. So it's obviously come from the rough endoplasmic reticulum. And this protein ends up here in this cistern. And that cistern has some specific composition of proteins when it's in this position. And that cistern is going to do something biologically magical to that protein. It's going to change it in some way. But once it's made all the changes that it needs to, instead of the protein moving from this cistern to this cistern, this whole cistern is going to move up. So the cisterns are actually what's moving in this model. So this cistern moves up to that position. And you can think about it just like this wave that's moving along the surface of the cistern. And each of those waves is carrying a protein in it. What's really cool about this model, though, is as that cistern moves, its protein composition changes. So it'll have a unique composition of proteins in position 1, and then the cistern will move to position 2, and its composition of proteins will change. And then that protein will get manipulated in that position. And then that cistern is going to move up here, and then to this position, and then finally to that position. So here, the salient point, the primary difference between the static cistern model and the cisternal maturation model is that the cisterns are moving, right, in the cisternal maturation model. It's not the product that's moving between cisterns. The actual cistern holds onto that protein and it moves across the surface of the Golgi apparatus until eventually that protein is completed and it buds off in a vesicle and goes somewhere else. Okay, does that make sense to everyone? If I asked you a short answer item on the exam, in a short answer item on the exam I asked you, explain the difference between the cisternal maturation model and the static cistern model. You could do that? Okay, yeah. I would be able to do that if I was you.
On the static, in orange, the vesicle, is that also a protein that is moving, or is it just a different product?
So it could be any product. Here I have it doing protein.
Okay.
But yeah, it could
It
be.
doesn't have to be protein specifically?
Yeah, the Golgi is able to manipulate any number of things, proteins, fats, sugars.
And that's the same for the cistern maturation model, if you do the maturations for any type of
Correct.
product.
Yep. Other questions? Yeah, please, Linda.
So the first one, is that the static cistern model, and the second
It
one
is.
is the, okay.
Cisternal maturation model. Yeah. So the question is, which of these two is right? And the answer is neither, really. I mean, it's probably some combination of the two. But this one, the cisternal maturation model, is more correct than the static.